Hello students. So today's topic is media preparation and sterilization. As you all are aware, the culture media consists of chemicals which supports the growth of microorganisms or culture. Microbes can be used the nutrients of culture media as their food and is necessary for cultivating them in vitro. So, what are the characteristics of the medium? So, medium should be neither too be acidic or not too alkaline. It should contain all kinds of nutrients in suitable amounts. So, a medium is sterilized before usage in the lab. Sterilization method includes autoclaving, dry heat, filtration, UV exposure, etc. So, the culture is a part of specimen grow in culture media. So, culture media can be defined as it is a medium, liquid, or can be solid that contains nutrients to grow bacteria in vitro. Because sometimes we cannot identify with microscopically examination, and sometimes we do culture for antibiotic sensitivity also. So, what are the properties of the media? It can support the growth of the bacteria. It should be nutritive. It have suitable pH. It has suitable temperature. Like for bacteria, grows at 37 degrees Celsius. There are many forms of culture. So we are discussing like solid culture, that is agar. It is a broth plus agar, and it is prepared by adding solidifying agent 1.5 to 3% of agar, and it is mainly prepared in petri dish but also in tubes and slopes. For example, like blood agar, chocolate agar, McConkey agar, etc. Semi-solid agar, which is also known as soft agar, it contains small amount of agar, that is 0.5 to 0.7%, used to check for the motility and also used as a transport media for fragile organisms. Liquid broth, it is mostly used for biochemical test, blood culture or broth culture. Growth of bacteria is shown by turbidity in the medium, for example nutrient broth or selenite F broth. So what are the properties of agar? It is a white like gelatin, and it melts at 97 degrees Celsius and solidified at 37 degrees Celsius. It comes as a solid powder form, and then we can add water to it and it forms a liquid medium. So what are the types of culture media? Simple culture media are that contains the basic nutrient that supports the growth of the bacteria. For example, nutrient broth, agar medium, peptone water, etc. Enriched culture media, these are the media which enriched with whole blood agar or lysed blood, which is heated to 80 degrees centigrade. For example, chocolate agar is also can be included in this media. Selective media, it is a media which contains substances that prevent or slow the growth of the microorganisms other than the bacteria for which the media is being prepared. For example, eosin methylene blue or triple sugar iron agar TSI. Differential media, it contains indicator dyes, etc. For example, McConkey agar, which contains neutral red and used to differentiate lactose fermenter and non-lactose fermenter. Some common media we can see, that is chocolate agar, which is prepared by heating blood to 95 degrees Celsius. McConkey agar, it is an inhibitory and differential medium used to distinguish lactose fermenting gram negative organism from non fermentation. Crystal violet or bile salts and neutral red are inhibitor agents for this. And there are also several medias like mannitol salt agar, MSA, Mueller Hinton agar, salmonella agar, triple sugar iron agar, which is the key medium for the use in the beginning, the identification of the gram-negative bacilli of the entire group. Now the media sterilization, it is done with the two process, autoclave and pressure cooker. First, while using the autoclave, the wet setting for sterilizing liquids like flask, bottles, and the use of dry setting when sterilizing empty containers. All the media should be sterilized for minimum about 45 minutes at high temperature and pressure. Remember not to tighten the cap or seal on any container. Like this is, you can see that these are the pictures of how to sterilize, and this is the picture of the autoclave, how the sterilization is being done. This is the 15 lbps pressure is being set on the autoclave. Thank you so much.